G'day everyone and welcome to another BioBoost video. This one's going to be done in two parts. Firstly, talking about photorespiration and what that is and why it's an issue, and then some of the adaptations that C4 and CAM plants have to avoid photorespiration. So this video will assume that you know the light dependent and light independent reactions of photosynthesis, and hopefully you've familiarized yourself with that before watching this video. Okay, so essentially we know that Rubisco is an enzyme that has a very important role in photosynthesis in C3 plants and C4 and CAM plants of fixing carbon. Okay, so we get carbon dioxide in, but that doesn't matter unless we can actually use it. So what we do is we fix carbon by putting it through a sequence of reactions, and Rubisco is the enzyme that's responsible for that. Now, the interesting thing about Rubisco is that it can bind with both carbon dioxide and oxygen. Okay, so that's why in my drawing that I've got here, I have Rubisco as this green enzyme that has this active site that can either fit carbon dioxide, but it can also fit oxygen. Okay, now in normal scenarios, there is approximately an 80 times higher affinity for carbon dioxide. Okay, so Rubisco wants to bind with carbon dioxide. If it can, it will. Okay, so in normal circumstances, photorespiration doesn't occur because carbon dioxide binds with Rubisco. Now, obviously, this is assuming that we have our stomata open and carbon dioxide is coming in. Okay. Now, why photorespiration can be an issue in C3 plants is that if we get things like hot conditions happening where we need to be closing our stomata to prevent water loss, because remember, H2O is lost when the stomata are open. Okay, we need to close our stomata so we have no new carbon dioxide coming in. Now, if we have no way to fix carbon and store it for later use, then across the day, if it's a hot conditions, we're going to get this buildup of oxygen that's being created in our normal photosynthesis reactions. That's A, struggling to get out, but also B, is becoming high in concentration compared to carbon dioxide, which again is not coming into the plant because the stomata are closed. Now, what happens is that in hot conditions, the Rubisco also becomes more likely to bind with oxygen. So not only does the affinity increase in hot conditions, but the ratio of oxygen also increases. So now we get Rubisco binding with oxygen instead of carbon dioxide. And what happens then? We get no carbon fixed, and now, what do you know? Photosynthesis goes down dramatically. Okay, the efficiency goes down a lot because suddenly our Rubisco can't bind to carbon dioxide. We're getting no carbon that we can then use to make glucose, which remember has 6 carbons, 12 hydrogens, and 6 oxygens. We need those carbons to make glucose, to make that sugar, and we can't. Okay, so this is what photorespiration is. Rubisco binding with oxygen rather than to carbon dioxide. So, now that you understand photorespiration, we'll talk about the adaptations that C3 and C4 plants have. So, I'm going to look at C4 plants first, but one thing you should see about both of these processes is that both of them separate out the carbon fixation by either getting different parts of the cell to do it or by doing it at different times of the day. So that's how they get around the issue. So here we can see over on the left-hand side, this is a C3 plant. Remember, this is most of our normal plants that we know that exist in normal, moderate conditions. We can see that normally the stomata are open. We get oxygen and water leaving the plant through the open stomata so that we can get carbon dioxide into the plant, which binds to rubisco and goes through the Calvin cycle, and we get glucose being produced. Okay, that's in a normal situation. But as we've seen in the last part of the video, that can be prone to photorespiration if the stomata close. Okay, we would get this shutting off. So we would get all this oxygen staying inside the plant cell. We would get no new carbon dioxide coming in, and photorespiration could occur. Okay, compare that to our C4 plant. Okay, what happens is that carbon dioxide comes in and is first transferred into the mesophyll cells. Okay, what it does is it basically stores carbon dioxide as malic acid. Okay, now when it stores it as malic acid, it then pushes it through into the bundle sheath cells where our Calvin cycle takes place. Okay, so it happens in a different location. So as we can see here, our pump, our CO2 pump, concentrates CO2 near rubisco, because rubisco is not in the mesophyll cell, it's in this bundle sheath cell. Okay, so even if we get hot conditions, okay, we've got carbon dioxide stored as malic acid that can then be pushed forward into our bundle sheath cells, but also there's no chance of oxygen getting into the bundle sheath cell. Okay, because it goes to the mesophyll cell, doesn't matter. Okay, what we do is we push malate into the bundle sheath cell, and that's where we get our carbon dioxide, and we can have our photosynthesis as normal. Okay, so that's a pretty cool adaptation. Now, in our CAM plants, instead of doing it in different parts of the cell like we saw it in the C4 plants, we now do it as part of a different time of day. Okay, so I'll just move my little person. So I really like this picture because it shows you what's happening at a broader sense. So we can see this plant cell here, this large central vacuole, and our chloroplast around the side. Now here we see that zoomed in, so we can see our chloroplast here and our vacuole over here. And what we can see is this split into day and night. Okay, so let's talk about what happens during the day. So these are plants that live in really hot, arid conditions, so dry conditions, so things like cacti. Okay, now imagine if they had their stomata open during the day. Okay, harsh heat, wind, whatever it is, they're going to lose too much water and they're not going to be able to survive. So what they do differently is they get their carbon dioxide at night. They have their stomata open only at night, so they get all this carbon dioxide coming in. Now remember, in normal C3 plants, yeah, they can open them at night, but they have no sunlight to be able to carry out the photosynthesis reaction, so they can't store the CO2. But that's where CAM plants have worked out the way. Okay, so the same in C4 plants. I actually didn't mention it, but I will note it here, that PEP carboxylase, this enzyme here, okay, I'll just circle where that is. PEP carboxylase is an enzyme found in both C4 plants and CAM, which fixes carbon differently to rubisco. So PEP carboxylase stores CO2 as malic acid. So the same thing is happening in CAM plants as in C4 plants, but it's just happening instead at a different time of day compared to C4 plants where it happened in a different cell type. Okay, so in CAM plants, we store CO2 that comes in during the night as malic acid, which is stored in the vacuole and left there until during the day, where we now close our stomatas to prevent water loss. And even though we're not getting CO2 coming into the plant during the day, we have all this malic acid that we saved from the previous night. Okay, so that malic acid is then brought into the chloroplast and processed and broken down into our carbon dioxide, which joins the Calvin cycle as normal. Okay, so very similar, but just one's happening at a different time of day. This one's happening just in different parts of the cell. But PEP carboxylase makes that possible by storing it as malic acid. Okay. Now, the last thing I wanted to talk about is just that difference between Rubisco and PEP carboxylase. I've changed the shape of CO2 here to really drive home the point that Rubisco and C3 plants, okay, so I'll say this, Rubisco in C3 plants are at danger of photorespiration because oxygen can bind to Rubisco just as CO2 can bind to Rubisco. Okay, and remember, all three of them have Rubisco, but Rubisco exists in a different cell type in C4 plants and is used at a different time of day in CAM plants. Okay, they use PEP carboxylase, and notice this difference in active site. Okay, PEP carboxylase cannot bind with oxygen. So even if there is oxygen present, which there potentially would be, it can't bind with it. So it can't undergo photorespiration. Okay, there is no chance for this to bind with this. The active site doesn't fit. It can't bind with PEP carboxylase. So instead, we always get carbon dioxide binding with PEP carboxylase and being turned into malic acid, which it can then use as a source of carbon dioxide later. So in our C4 and CAM plants, our Rubisco is never coming across large amounts of oxygen. So it's always going to preferentially bind with carbon dioxide and be used as normal in the Calvin cycle. Okay, so I hope that really helps to understand C3, C4 plants and CAM plants and how they differ from our normal C3 plants.